Today, we're going to be taking an overview over our project on identifying the biodegradability of bioplastics based on renewable resources. I'm Eric.
And I'm Kushal. Together, we're Biotech Chemistry.
One of the things that sparked our ideas for this research project is the growing immense problems of global warming, pollution, and climate change. With new strains of plastics, we believe that this can produce a positive impact on the environment. This is why we're testing the biodegradability of bioplastics. So now, let's dive into our project, starting with the abstract. Many types of bioplastics are derived from renewable resources. These can include a variety of ingredients such as corn, tapioca, and sugarcane. These experiments are conducted to find the better biodegradability of bioplastics by efficiently varying ingredients and their combinations used to construct them. An overview of a common bioplastic called polylactic acid, or PLA, is used as a base reference, where some features of bioplastics are stated. Bioplastics have the potential to replace traditional oil-based plastics only if their properties are modified to accommodate better biodegradability and easier manufacturing. This research project will identify the biodegradability of bioplastics based on renewable resources.
Along with the abstract, we also have a hypothesis. Based on research and background knowledge, we hypothesized that traditional bio-based plastics will degrade within 3 to 6 months. This was later questioned as the chemical known as amylase enzyme was supposedly able to accelerate this process. Background research. Before this project, let's do some background research about PLA or just any bioplastics. Bioplastics like PLA are vital in reducing pollution and can help reduce the dependency that humans have on fossil resources. Bio-based plastics minimize the use of oil as they are made of bio-based products. PLA uses 65% less energy than producing standard oil-based plastics. It also produces 68% fewer greenhouse gases than oil-based plastics. The fire diamond shows that PLA poses no health hazards and is safe in the category of instability/reactivity, but poses minor dangers in flammability. Overall, PLA poses minor peril and is almost entirely safe. Polylactic acid is noted as a substitutive renewable polymer for the replacement of traditional petroleum-based plastics. Both hydrolysis and the consumption of bacterial or terminal degraders can biodegrade PLA. Terminal degraders include microorganisms. PLA is also soluble in solvents, benzene, tetrahydrofuran, and dioxins. Oxidegradable and photodegradable plastics are both degradable, but are oil-based plastics. It neither has bonds in its structure that can be weakened or broken down by sunlight, or contains a chemical additive that absorbs light, attacking the bonds and breaking them. However, this produces toxic waste, especially in the marine environment. Presently, the most significant use for PLA is packaging: plastic utensils, cups, containers, and plastic bottles. PLA is additionally used in the medical industry. For example, like a pin or a plate that helps set a bone in place. PLA will gradually break down in the human body using the process of hydrolysis. An oxygen molecule combining with the hydrogen molecule, the chemical reaction breaking them apart. When this happens, weight slowly shifts back onto the healing bone. Another utilization of polylactic acid in the medical side is the use of PLA sutures. Instead of conventional sutures, doctors could use biodegradable stitches and seams. These sutures have the same capabilities, besides that the fact they can be broken by the body. Although they break down around 60 days, it's beneficial for them to cancel a trip to the hospital.
A further application for PLA is that it can also be used as a tissue scaffold. The PLA can be molded, and the tissue cells are then introduced to the formation, and circles around the structure formed into a configuration such as an ear until it forms the desired portion. This can also be implanted inside the human body and may be especially useful because as the cells mature around the structure inside the human body, the hydrogen molecules, the hydrogen with water molecules in the body can break down the formation. Amylase enzyme and how they affect bioplastics with hydrolysis. Hydrolysis, hydro meaning water, adding lysis, which means breakdown, is a chemical reaction with water where molecules are split apart. This term is used broadly for elimination and fragmentation reactions in which water is part of the nucleophile, a chemical species that gives an electron pair to form a chemical bond. Hydrolysis is the opposite of condensation. It is a chemical reaction where water breaks down another compound and changes its mixture. In most instances of organic hydrolysis, it combines water with neutral molecules, while synthetic hydrolysis pairs water with ionic molecules, such as acids, salts, and bases. Amylase is a type of enzyme that accelerates the process of hydrolysis. It works to biodegrade bioplastics because they hydrolyze starches and convert them into simple sugars. This carbohydrate may include a molecule formed of two glucose molecules called maltose. Amylase also comes from two parts of the human body, salivary glands and the pancreas. These are called alpha amylases. Meanwhile, beta amylases appear in bacteria, yeast, and plants, and also mold. These enzymes break down bioplastics and starches similarly to alphas. Beta amylases are the primary components of a diastase mixture utilized in the elimination of starchy-sizing agents from fabrics and other materials. It aids in the transformation of grains such as cereal to fermentable sugars as well. Looking at this chart over here, the leading manufacturer of polylactic acid seems to be NatureWorks in production capacity and quantity. This chart was found on plasticinsights.com. Another chart on the global production capacities of bioplastics show from 2018 to 2024 the different types of plastics and also the forecast. This was found on European Bioplastics.
This is a picture of lactide polymerization. Lactide is a renewable and natural substance created from the lactide produced by the fermentation of sugar. Lactide is a monomer with high purity and is used in the synthesis of polylactic acid as a raw material. Materials. Common formula for bioplastics is 1 tablespoon of cornstarch, potato starch, or tapioca starch. You can also use rice starch. 4 tablespoons of water, 1 teaspoon of glycerin, and 1 teaspoon of vinegar. The starches tested are a part of a molecular chain of sugar that is the main ingredient in the bioplastic. Water loosens up the starches chain while the vinegar breaks them. Glycerin also helps the bioplastic polymer slip against each other to create a flexible plastic.
So now let's move on to the procedure of our bioplastic biodegradation experiment. All the bioplastic ingredients and materials are combined together using heat from a cooktop. The newly forming substance soon becomes a gel and thickens as it heats up. Bubbling will occur after some time and indicates that the bioplastic cooking is done. This substance is then left out to dry, hardening the bioplastic. Once fully dry, the bioplastic can begin its phase of testing. This starts with water to begin biodegradation. Amylase enzymes then added into the water, inducing the activation of hydrolysis in the bioplastic. It will be monitored carefully for a few days, measuring the percentage over time it takes to biodegrade. A fully intact control will be used to measure the percent biodegraded. Data will be gathered in Excel charts to measure the time it takes for the bioplastics to degrade with percentages. This procedure will identify the biodegradability of bioplastics based on renewable resources and properties.
Results. We conducted an experiment on the degradation of bioplastics using hydrolysis and analyzed enzyme. Unfortunately, the experiment with the bioplastics analyzed enzyme failed and did not work as expected. A combination and confusion with the type of analyzed enzyme occurred between the alpha and beta varieties. The current analyzed enzyme did not seem to visibly biodegrade the bioplastics. Another earlier experiment was conducted with the corn bioplastic in normal freshwater over a course of days. Results in the Excel sheet shown. The Excel sheet are shown here in the poster. Though we didn't continue this experiment, this experiment was overtaken by mold and could have had the potential to biodegrade. Here we have the raw plastic. For the raw plastic, this is a failed experiment for cornstarch. This is also a failed experiment for the tapioca starch. This is a successful control for cornstarch. And this is a successful control for the potato starch. Here we have a picture of the cornstarch and saltwater analyzed enzyme. Here you can see some degradation. Here we have cornstarch in freshwater. Here you can also see some degradation. Here we have a picture of potato starch in freshwater with analyzed enzyme. Here you can see little to no degradation. Same with this picture of the potato with analyzed enzyme into saltwater.
So now let's move to the final conclusion. These bioplastics are based on renewable resources, including ingredients like corn, tapioca, and sugarcane. These experiments are performed to identify an improved version of bioplastic biodegradability by efficiently varying ingredients and combinations used to construct them. We have done intensive research on the common bioplastic known as polylactic acid, or PLA, and it is used as a base reference for our experiments. Bioplastics can certainly substitute normal oil-based plastics only if their characteristics are changed for improved biodegradability. This research is developed to identify the most convenient and environmentally friendly bioplastic. Unfortunately, this experimental research did not work correctly because of an error with amylase enzyme. We did not have enough time to reconstruct this research and experiments, so this is our unconcluded project. Now at the end, one thing we can do to further advance our research is testing the bioplastics in a real-world environment. We can further investigate and analyze the bioplastics biodegradability. Thank you for watching. Goodbye.